We often talk about metabolic fuel like it's just gas in a tank.
Right.
It's this very mechanical, sort of sterile analogy. You put the nozzle in, fill it up, and the car goes. We look at carbohydrates and we see this, clean, high-octane racing fuel. It burns fast, it burns bright.
Yeah, that's the simple narrative.
Yeah.
Carbs are for speed. Then you've got fats.
And fats are that slow-burning, diesel-dense, efficient, keeps you going for miles and miles.
Right. Fats are for endurance. It's clean, it's simple, easy to get your head around.
But then there is the third macronutrient, the one we are all, frankly, obsessed with. We drink shakes of it. We base entire diets around it. Protein. And today, we are finding out that when it comes to actually burning protein for energy, it is not a clean fuel at all. It's actually kind of a hazardous material situation.
It absolutely is.
Yeah.
I mean, if carbohydrates are solar power and fats are a high-capacity battery, burning protein is a bit like burning coal in your living room fireplace.
Oh, that's a good analogy.
It produces heat, sure, but it also produces a lot of toxic smoke and ash that you have to deal with immediately. If you don't ventilate that smoke, you are in very big trouble.
And that smoke is nitrogen.
Exactly. Nitrogen, the very element that makes protein, the thing that allows it to fold into enzymes and muscle fibers. But it's also the element that makes it so dangerous to metabolize.
So, welcome to the deep dive. Today we are tackling the biochemistry of amino acid oxidation and the production of urea, drawing from Leninger's principles of biochemistry and cross-referencing that with the clinical insights from Harper's Illustrated Biochemistry.
Which is a really great pairing for this topic.
It really is. Our mission today is to figure out how the body takes, say, a steak or a lentil stew, strips it for parts, and somehow manages to diffuse the nitrogen bomb hiding inside every single amino acid without, killing us.
It is a high-stakes operation. And what makes this deep dive so fascinating is that contrast between our sources. We have Leninger, which gives us this beautiful systems-level logic, the accounting of energy, the flow of atoms. It explains the chemical mechanisms, the why and the how.
And then we have Harper.
And then we have Harper, which acts like the physician in the room. Harper tells us, okay, that's the theory. Now, here is exactly what it looks like when this system breaks down in a patient.
Which, spoiler alert, looks terrifying.
It is not pretty. The consequences of failing to handle nitrogen are immediate, severe, and as we'll see, typically neurological.
So before we jump in, a quick disclaimer for you listening. This is a conceptual map. We are going to visualize the machinery, the flow of nitrogen, and the logic of the liver. We aren't going to read out every single chemical structure or reaction rate.
No, absolutely not.
So use this discussion as your scaffold. When you go back to read the source material, the equations will make a lot more sense because you'll understand the story behind them.
Precisely. We are here to connect the dots, not just count them.
Okay, so let's unpack the basic premise. Why do we call protein the fuel with a cost? I mean, I eat a chicken breast, I feel fine. I don't feel like I'm handling toxic waste.
You feel fine because your liver is working overtime. See, the fundamental problem with amino acids is storage. Think about sugar. If you eat too much sugar, what happens?
It gets stored as glycogen in the liver or muscles, or, turned into fat if you really overdo it.
Right. We have a massive capacity to store lipids in adipocytes. We have a decent, if limited, capacity to store carbohydrates as glycogen, but we cannot store amino acids. There is no protein tank in the body.
Wait, really? I always kind of thought my muscles were my protein tank. That's why people bulk up, right? To store protein.
That's a very common misconception. Your muscles are functional tissue. They're structural. They're contractile. They are enzymes and filaments doing actual work. They're not storage depots waiting to be tapped like a pantry.
Ah.
If you are breaking down muscle for energy, you are literally eating your own walls to feed the fireplace. You are degrading your functional capacity.
That is a grim image, eating the walls to keep the house warm.
It is. So amino acids in the body, they effectively have two paths. They are either used immediately for synthesis to build new proteins, enzymes, DNA, neurotransmitters, or if they aren't needed for that, they must be degraded. You use it or you lose it.
And the losing it part is where the oxidation comes in. This is where we burn it for fuel.
Correct. And Leninger is very specific about this. There are three main circumstances where we actually burn amino acids. Because remember, it's not the body's first choice.
Okay, let's list them. Scenario 1.
Normal cellular turnover. Your body is constantly recycling. It's not just about what you ate for lunch. Every single protein in your body has a shelf life. Old proteins break down, new ones are built. Not all the amino acids get reused perfectly, so the surplus gets burned. This is just the background hum of keeping the lights on.
So even if I'm fasting, I'm still burning some amount of protein just from recycling myself.
Exactly. Scenario 2 is dietary excess. This is the carnivore diet problem, or just Thanksgiving dinner.
Yeah.
If you eat way more protein than you need for synthesis, you cannot store the rest.
Yeah.
As we said, there's no tank.
Yeah.
So the body has no choice but to burn the surplus for energy or convert the carbon skeleton into fat. But to do either, it has to deal with the nitrogen first.
Got it. And scenario 3.
Starvation. Or uncontrolled diabetes. This is crisis mode. This is where carbohydrates are either unavailable, as in starvation, or the body can't see them because of lack of insulin, as in diabetes. The body gets desperate and starts breaking down its own functional tissue muscle to stay alive.
So in all 3 cases, we're left with an amino acid, and it's basically a carbon skeleton, which looks like good fuel, attached to this amino group, the nitrogen.
And that is the problem. The carbon skeleton, great fuel. We can send that to the citric acid cycle, burn it up, make ATP. We love carbon. But that amino group, that nitrogen, that is the hot potato. You cannot just chop it off and let it float away.
Because free ammonia at NH3, or its protonated form, NH4+, is toxic.
Highly toxic.
Wow.
So step 1 of this whole disposal process is what you call the nitrogen handoff. We need to move the nitrogen somewhere safe without letting it float free.
Right. And this process is called transamination. And the mechanism here is honestly fascinating. It's not just a simple swap. Harper describes it as a ping-pong mechanism.
The ping-pong bi-beam mechanism, to be technically precise.
I love that. Describe the game for us. How does this work mechanically?
Okay, let's zoom in on the enzyme. These are called aminotransferases. You might also know them as transaminases. Imagine the enzyme as a table. An amino acid, let's say alanine, walks up to the table holding the nitrogen ball.
Okay, alanine is holding the ball, ready to play.
Alanine docks into the active site of the enzyme. Now, waiting inside that active site is a very important cofactor called PLP, pyridoxal phosphate. This is actually the active form of vitamin B6.
Ah, so that is why vitamin B6 is essential. It's the other player at the table.
It's the glove. It's what catches the ball. PLP has a very specific chemical structure. It has an aldehyde group, a carbon double bonded to an oxygen. That aldehyde is the bait.
The bait for what?
For the nitrogen on the alanine. The nitrogen's lone pair of electrons attacks that aldehyde carbon. It kicks out a water molecule and forms a new double bond between the nitrogen and the PLP. This structure is called a Schiff base.
A Schiff base. I remember that term from organic chemistry nightmares. It's a carbon-nitrogen double bond.
Exactly. And the reason biology uses a Schiff base here is that it's what we call an electron sink. It allows the enzyme to play a trick with the electrons. It pulls electron density away from the bond connecting the nitrogen to the rest of the alanine. And with little chemical rearrangement, snap. The carbon skeleton of the alanine falls off.
So the alanine leaves the table.
Yes, but it leaves as an alpha-keto acid, in this case pyruvate. It has lost its nitrogen. It's free to go be fuel now. But the enzyme is now holding the bag. The PLP has become PMP, pyridoxamine phosphate. It's holding the nitrogen.
That's the ping.
That's the ping.
What's the pong?
The pong is the second half. Another molecule walks up to the table. This is almost always alpha-ketoglutarate.
Alpha-ketoglutarate. That's a Krebs cycle intermediate, isn't it? One of the central hubs of metabolism.
It is. It's ubiquitous. It walks into the active site, and it does the reverse reaction. It forms its own Schiff base with the nitrogen that the enzyme is holding. The enzyme then passes the nitrogen on to the alpha-ketoglutarate.
And when alpha-ketoglutarate takes the nitrogen, what does it become?
It becomes L-glutamate.
This brings us to what the outline calls the funnel effect. This blew my mind when I read it. We have 20 different common amino acids, right? Do we have 20 different disposal systems?
No, that would be incredibly inefficient. Biology loves to standardize no matter which amino acid you start with, well, almost any of them. The nitrogen eventually gets passed to alpha-ketoglutarate to become glutamate. It is like all these small tributaries flowing into one big river. Glutamate is the central reservoir for nitrogen in the cell.
And this happens via those enzymes you mentioned, the aminotransferases. And I recognize these names from my last physical exam, ALT and AST.
Yes, alanine aminotransferase, ALT, and aspartate aminotransferase, AST. Very famous enzymes.
I always see these on blood work under liver function. I just assumed they were, general liver juices. I didn't realize they were these specific ping pong players.
They are the workers doing this nitrogen swapping inside the liver cells, the hepatocytes. The reason they appear on your blood test is pure clinical relevance. If you see high levels of ALT or AST floating in your blood, it implies that the liver cells have burst open.
Oh, So it's not that the enzymes are working in the blood. That's not their job site?
No, absolutely not. They should be inside the cell. If they're in the blood, it means the cell membrane has been compromised.
Yeah.
They're leaking their internal machinery out into the street. So high ALT isn't just a random number. It is evidence of cellular looting or necrosis hepatitis, alcohol damage, drug toxicity.
That makes the blood test result much more vivid. It's a debris field. Okay, so normally these enzymes stay inside the cells, funneling all that nitrogen onto glutamate. But this is happening all over the body, right? In my biceps, in my brain.
Correct. Protein turnover happens everywhere.
But the main processing plant, the urea cycle, is only in the liver. So we have a big logistics problem. We have a toxic cargo being generated in the tissues, muscle, brain, and we need to get it to the liver without poisoning the bloodstream along the way.
This is part 2, transporting the hazardous cargo. You cannot just dump free ammonia into the blood. It is far too dangerous.
Why? I mean, what actually happens if ammonia levels rise in the blood?
It is catastrophic for the central nervous system. We will get into the specific molecular mechanism of why in a bit, but clinically. You're talking about tremors, slurred speech, blurred vision, eventually coma and death. The margin of error is tiny. Normal blood ammonia is incredibly low single-digit micromolar concentrations, so the body has to use safe carriers.
Let's talk about solution A, glutamine. This is the general purpose truck.
Glutamine. This is the general transport solution. Let's look at the brain. Your brain is constantly breaking down neurotransmitters and proteins. It generates free ammonia. That ammonia is incredibly dangerous to neurons.
The brain has to mask it fast.
Right. It uses a very important enzyme called glutamine synthetase. This is a 2-step reaction. First, it takes glutamate, which, remember, already has 1 nitrogen, and it activates it with ATP.
So we're spending energy here.
Yes, and ATP is hydrolyzed to ADP. Then it shoves a free ammonia molecule onto that activated glutamate. Now you have a molecule with 2 nitrogens. That is glutamine.
It's like double bagging the trash to make sure nothing leaks.
It's a perfect analogy. Glutamine is neutral, non-toxic, and it floats easily through the blood. In fact, glutamine is the most abundant amino acid in your bloodstream precisely because it's doing this transport job. It travels to the liver and kidneys, where the nitrogen can be unloaded safely inside the mitochondria.
Okay, so that's the general solution. But the muscles have their own special deal, don't they? The glucose-alanine cycle.
This is one of my favorite cycles in all of biochemistry, because it shows how the liver acts as the altruistic organ for the rest of the body.
Break it down for us. I've just done a heavy set of squats. My muscles are breaking down some protein for repair and energy.
Right. Your muscles are working hard. They're running glycolysis, burning glucose furiously. The byproduct of glycolysis is pyruvate. But at the same time, the muscle is breaking down protein, generating toxic nitrogen. It needs to get rid of that nitrogen, but it also desperately needs more glucose to keep working.
So it tries to solve both problems at once.
It does. The muscle uses ALT-alanine aminotransferase. It takes the nitrogen, which has been funneled onto glutamate, and sticks it onto the pyruvate. Pyruvate plus a nitrogen group equals alanine.
And alanine is just another amino acid.
It is, and it's safe to travel in the blood. So alanine travels through the blood to the liver. The liver takes the alanine inside, runs the reaction in reverse, strips off the nitrogen to deal with it via the urea cycle, and what is left?
Pyruvate again.
Pyruvate. Now here's the magic. The liver takes that pyruvate and turns it back into glucose via gluconeogenesis. It then releases that fresh glucose back into the blood for the muscle to use.
That is a sweet deal for the muscle. It's like, here's my toxic trash. Could you please send it back to me as sandwiches?
It is incredible. The muscle gets to dump its waste and gets fresh fuel in return. But look at the energetic cost. The liver is spending its own ATP to turn that pyruvate back into glucose. Gluconeogenesis is expensive. It costs 6 high-energy bonds to make 1 glucose.
Well, the muscle only gets 2 ATP net from burning that glucose in glycolysis.
So the liver is operating at a massive net loss. Leninger emphasizes this point. This cycle imposes the energetic burden of gluconeogenesis on the liver, sparing the muscle's ATP for contraction. The liver effectively subsidizes your workout.
The liver really is the unsung hero of metabolism. It takes the toxic hit and it pays the energy bill.
It is the metabolic bedrock.
Okay, so whether it comes by glutamine or alanine, all this nitrogen eventually arrives at the liver.
The processing plant.
And now we have to finally open the bags. We have to liberate the ammonia.
This is part 3, deamination. We are deep in the liver mitochondria now. The glutamine and alanine have dropped off their nitrogen loads onto glutamate. So now we have a big pool of glutamate in the liver mitochondria.
And we need to get the nitrogen off the glutamate so we can finally turn it into urea.
Correct. The key enzyme here is L-glutamate dehydrogenase. It performs what's called oxidative deamination.
What's unique about this enzyme? Leninger makes a big deal about its cofactor usage.
It is weird. Most dehydrogenases are picky. They use either NAD+ or NADP+. Glutamate dehydrogenase can use either one. It's the only enzyme we know of that has this dual capability. It strips the nitrogen off glutamate, releasing it as free ammonium, NH2SO4, and regenerating our old friend alpha-ketoglutarate, which can go pick up more nitrogen.
But now we have the problem we were avoiding this whole time. We have free ammonia right there in the liver mitochondria. Before we turn it into urea, I want to pause on the toxicity. The outline mentions a thought experiment from Leninger about why ammonia is actually toxic.
Yes, this is crucial because for a long time, people just thought, oh, it's a base that changes the pH. But the toxicity happens at concentrations way too low to affect blood pH significantly. It's a metabolic toxicity.
So walk us through the disaster scenario. The liver fails, ammonia rises, it hits the brain. What is the molecular mechanism of the coma?
It's a cascade. Harper and Leninger outlined a specific vicious cycle in the brain. Step 1. Ammonia crosses the blood-brain barrier easily. It's a small, uncharged molecule, so it basically walks right through the walls.
And once it's inside the brain cells, specifically the astrocytes, the support cells, they try to defend themselves.
They panic. They grab the nearest glutamate molecule and say, quick, attach this ammonia to it. They use that enzyme we mentioned earlier, glutamine synthetase, to turn glutamate into glutamine.
Which sounds smart, right? Glutamine is neutral. It's safe.
It's safe chemically, but not physically. This is the osmotic theory of ammonia toxicity. Glutamine is an osmolyte. It loves water. As glutamine piles up inside the astrocytes, it starts sucking water in from the blood by osmosis.
The cells start to inflate.
They swell up like balloons. But the brain is in a fixed box, the skull. There is no room for swelling. The intracranial pressure goes up, crushing the blood vessels. You get cerebral edema and ischemia.
So the brain effectively strangles itself, trying to hide the ammonia.
That's the physical damage. Then there's the metabolic damage. Remember, to make all that glutamine, you used up glutamate. And to replenish that glutamate, the cell pulls alpha-ketoglutarate out of the Krebs cycle.
And alpha-ketoglutarate is a critical part of the fuel line.
Exactly. If you deplete alpha-ketoglutarate to mop up ammonia, the Krebs cycle in your neurons stalls. The engine dies.
Right.
Energy failure. ATP levels plummet.
And Leninger also mentioned something about potassium ions.
Yes, this is a more subtle point. The ammonium ion, NH4 plus I, is very similar in size to the potassium ion, K plus I. It can compete for potassium transporters, like the NKCC1 transporter. This disrupts the carefully balanced ion gradients across the neuronal membrane, which messes with the electrical firing of the neurons.
So it's a triple threat. You have energy failure, physical swelling from osmosis, and electrical short-circuiting.
It is. No wonder the body spends so much energy to avoid this. It puts the cost of the urea cycle into perspective. We're about to see that making urea is energetically expensive, but the alternative is brain death.
So let's get to the solution. Part 4, the urea cycle, the disposal plant.
This was actually the first metabolic cycle ever discovered by Hans Krebs and Kurt Henseleit in 1932. So before the famous Krebs cycle, there was the urea cycle.
There is a geography to this, right? It isn't all happening in one pot.
Correct. It starts in the mitochondria, the high-security area where the free ammonia is generated, and then it moves out to the cytosol. This compartmentalization is key to regulation.
Okay, walk us through step 1, the pacemaker.
We are in the mitochondrial matrix. We have this dangerous free ammonia. We have bicarbonate, which is basically dissolved CO2 from our breathing. We need to fuse them together. This requires the enzyme carbamoyl phosphate synthetase SF, or CPSI.
And this is the rate-limiting step, the main control point.
This is the choke point. And let's look at the bill for this step. It requires 2 ATP molecules.
2. Usually 1 ATP is enough to activate a reaction. Why 2?
Because ammonia and bicarbonate really hate each other. They do not want to bond, so we have to bribe them heavily. It's a 2-step activation. The first ATP phosphorylates the bicarbonate to make it reactive. That allows the ammonia to attack it, forming a molecule called carbonate.
Okay, so we have carbamate.
But carbonate is still pretty stable. So we use the second ATP to slap another phosphate onto the carbonate. Now we have carbamoyl phosphate, and that molecule is just vibrating with energy. It's desperate to get rid of that phosphate group.
So we've essentially primed the explosive.
Exactly. We've invested high-energy currency to create a molecule unstable enough to enter the cycle proper.
The outline mentions a special ticket required for this enzyme to even work.
Yes, N-acetylglutamate. This is a crucial regulatory concept.
Mm.
CPSI is a very resource-intensive machine. You only want it turned on when you actually have excess nitrogen to deal with. So the presence of N-acetylglutamate acts as the on switch. It's an allosteric activator.
Where does N-acetylglutamate come from?
It's made from acetyl-CoA and glutamate. Think about the logic. If you have lots of glutamate, that means lots of nitrogen breakdown is happening. If you have lots of acetyl-CoA, that means metabolism is running high. When both are high, levels of N-acetylglutamate rise, turning on the urea cycle. It's a perfect feed-forward activation.
And if you don't have that switch, if that molecule is missing,
You die of ammonia toxicity. Even if the CPSI enzyme itself is perfectly fine, if the switch isn't flipped, the cycle doesn't start. This is one of the rare genetic disorders we will touch on later.
okay, so we've made our high-energy carbamoyl phosphate, step 2.
We need a carrier, a molecule that acts like the truck chassis to carry this nitrogen through the cycle. That molecule is ornithine.
Ornithine. That isn't one of the standard 20 amino acids we learn in Biology 101.
No, it is not used to build proteins. Its entire life, its destiny, is to work in this cycle. Carbamoyl phosphate fuses with ornithine to form a new molecule, citrulline. The phosphate falls off. That was the energy source driving the bond.
And citrulline can leave the mitochondria.
Yes, citrulline is the export model. It has a specific transporter that moves it out of the mitochondria into the cytosol. Now we are on the general factory floor.
So now we're in step 3. Out in the cytosol, we have one nitrogen on board, the one from the ammonia, but urea has 2 nitrogens. Where does the second one come from?
From our old friend aspartate. Aspartate enters the cycle here.
Aspartate enters the chat.
Citrulline plus aspartate combines to form a massive molecule called arginasuccinate. This reaction is catalyzed by arginasuccinate synthetase.
And what's the energy cost here?
Another ATP. But it's not just ATP to ADP. It's ATP being split into AMP and pyrophosphate.
Which is energetically equivalent to spending 2 ATPs, right? Because the cell has to immediately break down that pyrophosphate and then spend another ATP to recharge AMP back to ADP.
Exactly. So if you're keeping score, we spent 2 ATP in step 1, and essentially 2 ATP equivalents here. We are now 4 high-energy bonds in the whole.
That is incredibly expensive just to get rid of some nitrogen.
It is, which tells you how dangerous the alternative is. So, step 4. We have this big, clunky arginasuccinate molecule.
What happens to it?
We cleave it using arginasuccinate lyase. We keep the part with all the nitrogens, that becomes arginine, and we cut loose the rest of the carbon skeleton as fumarate.
Arginine. Now that is one of the standard amino acids.
It is. In fact, this is how our body synthesizes arginine. But here in the liver, its destiny is to be chopped up almost immediately.
So step 5, the finale.
The enzyme arginase adds a water molecule, a hydrolysis reaction. It snips off the end of the arginine molecule. That little snipped off piece is urea. Harmless, water-soluble urea. It diffuses into the blood, goes to the kidneys, and is excreted in the urine.
And what is left of the arginine after you snip off the urea?
Ornithine. The truck chassis is empty again. It gets transported back into the mitochondria to pick up another load of carbamoyl phosphate. The cycle is complete.
Elegant. But let's go back to that piece we cut off in step 4. Fumarate. That sounds very familiar.
It should. Fumarate is an intermediate in the citric acid cycle, the Krebs cycle.
So the urea cycle in the cytosol spits out a piece of the Krebs cycle, which runs in the mitochondria.
Exactly. Leininger calls this the Krebs bicycle.
The bicycle. I like that.
Imagine 2 wheels connected by an axle. One wheel is the urea cycle, the other is the citric acid cycle. The urea cycle produces fumarate. Fumarate in the cytosol is converted to malate. Malate can be transported back into the mitochondria.
And once malate is in the mitochondria,
It re-enters the Krebs cycle. The enzyme malate dehydrogenase turns it into oxaloacetate. And crucially, that reaction produces 1 molecule of NADH.
NADH, that's electron transport fuel. That makes ATP.
Yes. When that NADH is oxidized in the electron transport chain, it produces about 2.5 APP. Wait, so let me do the math here. The urea cycle costs 4 high-energy bonds, so 4 ATP. But this bicycle connection generates about 2.5 ATP worth of NADH. So the net cost is effectively reduced to about 1.5 ATP per molecule of urea.
So the body is recouping some of the cost of detox.
It is. The bicycle connection helps generate enough energy to offset some of the massive cost of the urea cycle. It reduces the energetic penalty of detoxifying ammonia. It is a brilliant example of system integration. The waste of one cycle is used to help fuel the other.
That is amazing design. It's so efficient.
Nothing is wasted in biochemistry if it can be helped.
So we have built this beautiful, intricate machine, But as Harper reminds us, machines can break. This brings us to part 5, clinical relevance and metabolic disorders.
This is where we shift from the lab bench to the bedside. Urea cycle disorders, or UCDs.
Harper says these are individually rare, but as a group, they present a nightmare scenario for a pediatrician. What does a classic case look like?
Picture this. You have a neonate, a newborn baby. Full term, looks perfectly healthy. APGAR scores are 10 out of 10.
Why are they normal at birth?
Because while they were in the womb, the placenta was their dialysis machine. The mother's liver was doing all the work for them. Her blood cleaned their blood.
But then the umbilical cord is cut.
They are on their own. They start feeding. Breast milk or formula, it has protein. They digest it, absorb the amino acids, and suddenly they have a nitrogen load. But they have a defect in one of these 5 urea cycle enzymes.
Harper mentions a characteristic symptom-free interval.
Yes, usually about 24 to 48 hours. Then, as ammonia builds up, the baby becomes lethargic. They stop feeding. They might have vomiting. They often develop hypothermia, and then breathing issues, apnea, where they stop breathing, or hyperventilation.
Hyperventilation? That
Yep.
seems odd.
Yes, it's a central effect of the high ammonia on the brainstem. They blow off CO2, which leads to respiratory alkalosis. If you don't catch it at this stage, they slip into a coma and die of that cerebral edema we talked about.
So let's play detective. You're the doctor. You have a baby with sky-high ammonia. How do you know which enzyme is broken?
You follow the metabolites. You look at what's building up behind the dam. Let's look at the specific defects. Hyperammonemia type 1 is a defect in the very first step, CPSI or the switch, and acetylglutamate synthase.
If that first step is broken, you can't make citrulline.
Exactly. So if you measure the blood, ammonia is sky high, but citrulline is undetectable. The factory line stopped right at the front door.
Okay, what about hyperammonemia type 2?
That's ornithine transcarbamylase, or OTC, deficiency. This is the most common one, and it's an X-linked disorder, so it mostly affects boys. Here, you can make carbamoyl phosphate, but you can't attach it to ornithine.
So carbamoyl phosphate builds up?
Yes, and it has nowhere to go, so it spills out of the mitochondria and gets shunted into a different pathway, the pyrimidine synthesis pathway. This creates a buildup as a molecule called orotic acid. So if you see high ammonia, low citrulline, but high orotic acid in the urine, that's the fingerprint for OTC deficiency.
That is incredible biochemical logic. And then there is citrullinemia.
As the name implies, that's a defect in arginosuccinate synthetase. In this case, you can make citrulline just fine, but you can't process it further. So citrulline builds up to massive levels in the blood.
So the diagnostic clue is high ammonia plus extremely high citrulline equals citrullinemia.
Precisely. It's biochemical troubleshooting at its finest.
How do you treat this? I mean, we established that we need protein to live.
Huh.
But protein is literally poison to these kids.
That is the ultimate catch-22. You absolutely must restrict dietary protein to minimize the nitrogen load. But you cannot eliminate it. Humans cannot synthesize about half of the amino acids, the essential amino acids. If you don't eat them, you can't build your own body proteins. You will die of wasting.
So it is a constant tightrope walk.
A very precise one. You titrate protein intake exactly to the growth needs of the child, not a gram more. And you use drugs like sodium phenylbutyrate, which acts as a nitrogen scavenger. It binds to glutamine and forces it to be excreted in the urine, basically creating an alternative exit door for the nitrogen that bypasses the urea cycle entirely.
So you're providing a chemical workaround.
Exactly. And interestingly, you often supplement with arginine.
Why arginine?
Well, remember, the cycle makes arginine. If the cycle is broken before that step, the patient can't make their own arginine, and it becomes essential. Plus, high levels of arginine can sometimes help drive the remaining parts of the cycle forward by mass action.
It sounds like an incredibly difficult condition to manage.
It is, but thanks to modern newborn screening programs, we can catch it early. Harper mentions tandem mass spectrometry. We can detect these abnormal metabolites in a single drop of blood from a heel stick within days of birth. We can start treatment before the brain swells.
Saving brains before the damage is done.
That's the whole game.
Okay, we have dealt with the nitrogen, we have flushed it away as urea, but we still have the rest of the amino acid left over. The carbon skeleton. This is part 6.
This is the payoff. We have paid the nitrogen tax, and now we get to keep the profit, which is the carbon.
And the profit takes 2 forms, ketogenic or glucogenic. This distinction used to really confuse me in medical school.
It's all about where the skeleton enters the metabolic map. It's a question of fate.
Ketogenic means it can turn into ketone bodies or fat.
Essentially, it breaks down into acetyl-CoA or acetoacetyl-CoA. Now, here is a hard rule in animal metabolism. You cannot turn acetyl-CoA back into glucose. The enzyme pyruvate dehydrogenase is a one-way street. It's irreversible.
So if an amino acid becomes acetyl-CoA, there's no going back to sugar.
No going back. It can be burned for energy in the Krebs cycle, or it can be made into ketone bodies or fat. It cannot be used to sustain blood sugar.
And which amino acids are strictly ketogenic? Leninger has a specific list.
There are only two purely ketogenic amino acids, leucine and lysine, the L amino acids. If you were starving and you ate a bowl of pure leucine, you would still die of hypoglycemia. You cannot make sugar out of it.
That is surprising. I feel like leucine is always sold as this magical energy-boosting supplement for athletes.
It is great for signaling of muscle growth. It activates a pathway called mTOR, but as a glucogenic fuel source for your brain, useless.
Okay, so the rest are glucogenic.
Most are either purely glucogenic or mixed, meaning part of them becomes ketogenic and part becomes glucogenic. They degrade into pyruvate or intermediates of the Krebs cycle, like oxaloacetate or succinyl-CoA.
And since those are part of the cycle, the liver can run those intermediates backward up the chain to make fresh glucose.
Correct. This is why protein can stabilize blood sugar. During starvation, your liver strips the nitrogen off your muscle protein, takes the glucogenic skeletons like alanine, turns them into glucose through gluconeogenesis, and feeds your brain.
I want to zoom in on one specific pathway defect here because the name is so vivid. Maple syrup urine disease, MSUD.
Yes. A classic clinical pearl.
It sounds like a joke or maybe a breakfast mishap, but it's incredibly serious.
Very serious. It involves the three branched-chain amino acids, leucine, isoleucine, and valine.
The BCAAs.
Yeah.
The bodybuilder's favorite supplement.
Yes. Structurally, these are bulky.
Yeah.
They have these branched carbon chains. They need a special dedicated enzyme complex to break them down. The branched-chain alpha-keto acid dehydrogenase complex.
That is a mouthful.
It is. But structurally, this enzyme is practically a clone of the pyruvate dehydrogenase complex. It uses the exact same 5 cofactors: thiamine, lipoD, FAD, NAD, and coenzyme A, the famous tender loving care for Nancy mnemonic.
And if this enzyme is defective, you simply cannot break down these 3 amino acids.
So they build up, and their deaminated keto acid forms build up. And these specific keto acids have a sweet, distinctive smell.
Hence the name.
Hence, the urine and even the earwax of the affected infant smells like burnt sugar or maple syrup.
And the symptoms.
They're identical to the other metabolic crises we've discussed: severe neurological damage, ketoacidosis. The brain cannot handle the imbalance of these bulky amino acids. And again, the treatment is painstaking dietary restriction. You have to limit leucine, isoleucine, and valine, but you can't eliminate them because they're all essential for life.
It's the same tightrope as the urea cycle disorders.
The exact same tightrope, just a different biochemical mechanism.
And we should probably give a brief mention to the most famous one of all, PKU, phenylketonuria.
The one everyone learns about. This is a defect in phenylalanine hydroxylase. Normally, our bodies take the essential amino acid phenylalanine and convert it into another amino acid, tyrosine.
So we make tyrosine from phenylalanine.
Yes. But if that enzyme is broken, phenylalanine builds up and gets shunted down other pathways, creating neurotoxic byproducts. But here is the fascinating chemical quirk that Harper points out. Normally, tyrosine is considered non-essential because we can make it.
But if you can't make it,
tyrosine becomes a conditionally essential amino acid. You have to get it from your diet because your internal factory is broken. If you treat a PKU patient by just cutting out phenylalanine from their diet, they will become deficient in tyrosine, which is needed to make things like dopamine and melanin.
It fundamentally changes the nutritional requirements of that human being.
Fundamentally.
So let's try to synthesize all this. We have gone from the ping pong of transamination to the bicycle of the urea cycle. What's the big picture?
I think the big takeaway is the duality of protein. We revere it as this essential building block. We think of it as good food. But metabolically, it is a burden. It is a hazardous fuel.
It forces the liver to be this chemical hero.
Absolutely. The liver accepts the nitrogen from the muscle via alanine, from the brain via glutamine, and from the gut. It centralizes the toxicity. It acts as the hazardous waste processing facility for the entire body. It spends its own expensive ATP to neutralize this threat so the brain can keep thinking and the muscles can keep moving.
It's the ultimate metabolic altruism. The liver pays the bill for everyone else.
It does. And it reminds us that our metabolism is designed for integration and survival, not just simple storage. The complexity of the urea cycle, the way it connects the mitochondria, the cytosol, the Krebs cycle, It shows how biology repurposes everything. Even the exhaust pipe of the urea cycle, that fumarate, is plumbed right back into the engine to squeeze out a little more power.
Here is a final thought for you to chew on, listener. We talked about how expensive this is. 4 high-energy bonds per molecule of urea. That is a huge energy tax.
A huge tax.
It makes you wonder about the evolutionary pressure. Why did we stick with this system? Why not just excrete ammonia directly like fish do? Fish don't have urea cycles, do they?
Most don't. Fish are ammonotelic. They live in an infinite ocean of water. They can dilute the ammonia immediately. They just let it diffuse out of their gills. The concentration never gets high enough to be toxic.
But we don't have that luxury.
We left the ocean. We are terrestrial. We carry our own little ocean inside us, our blood. But we have a limited amount of water. We cannot afford to urinate 50 gallons a day just to dilute ammonia down to safe levels. We would desiccate and die.
So we needed a solid waste product, or at least a more concentrated one.
We need a soluble, concentrated, non-toxic one. Birds went even further. They make uric acid, which is almost a paste, to save even more water and weight for flying. We mammals chose urea. It costs a lot of energy to make, but it allows us to conserve precious water.
So the urea cycle is actually the price of living on land.
It is the metabolic cost of leaving the ocean. Without the urea cycle, terrestrial life as we know it would be impossible. We pay 4 ATP for the privilege of not being fish.
Wow. Next time you eat a burger, remember, you are paying the nitrogen tax for the privilege of walking on dry land.
I will never look at a burger or my liver the same way again.
Thanks for diving in with us.
It was a pleasure.
We'll see you on the next Deep Dive.